Hello friends, welcome back to the next module of this chapter. Today in this module we are discussing two important structures of the nephron, that is the Malpighian body and the renal tubule. Now let's start first with Malpighian body. In the previous module we have said that Malpighian body is also called as renal corpuscle, which can be also termed as Pygmalion corpuscle. And in the previous module, we also discussed that Malpighian body is made up of two important structures, that is the Bowman's capsule, and inside the Bowman's capsule there is glomerulus. So Bowman's capsule plus glomerulus is collectively termed as Malpighian body. Now let's see the Bowman's capsule. Now Bowman's capsule is a double-walled cup-like structure which is having the proximal end blind. And if you see the outer covering of the Bowman's capsule, as I told you, it's a cup-like structure, so it has two coverings, the outer covering and the inner covering. The outer covering of the Bowman's capsule is termed as the outer parietal layer, which is formed by single layer of epithelial cells called as squamous epithelium. The inner layer is called as the visceral layer, and the inner layer is called as visceral layer, which is also formed by single layer of epithelial cells called as squamous epithelium. But along with the simple squamous epithelium of the inner visceral layer, there is also presence of specialized cells which are called as podocytes. Now the function of the podocytes we will discuss in the later modules. And the space which is present in between the outer parietal layer and the inner visceral layer is called as the urinary space or the Bowman's space. So as the blood gets filtered from the glomerulus, the urine would be collected inside the urinary space or the Bowman's space. Now let's see what is glomerulus. So here you can see inside the Bowman's capsule, you can see a multiple junction or tuft of capillaries. Around tuft of 50 capillaries is forming your glomerulus. So glomerulus is nothing but tuft of around 50 capillaries. How are these 50 capillaries formed? These are formed due to the division of afferent arteriole. In the previous model we studied that inside the Bowman's capsule what enters? The afferent arteriole, and the afferent arteriole divides to form the glomerular capillaries. After this formation of glomerulus, all the capillaries rejoin to form and exit the Bowman's capsule as efferent arteriole. And the diameter of afferent arteriole is larger than the efferent arteriole. You can appreciate that in the diagram, and this difference in the diameter between the afferent and efferent arteriole is very important for the filtration of blood. That would be also discussed in the later modules when we'll discuss the formation of urine. Now let's see the renal tubule. In the previous module we discussed that renal tubule is formed by three parts. The proximal tubule, which is also called as PCT or proximal convoluted tubule, the U-shaped tubule. The proximal tubule continues into a U-shaped tubule called as loop of Henle, and the loop of Henle then continues as the distal tubule, which is also called as the distal convoluted tubule. Now let's see each part individually. Now let's see first the PCT, that is the proximal convoluted tubule. Now you can see the PCT lies in the cortex, and it is highly coiled, present in the cortical region, and inside the proximal convoluted tubule you can see cuboidal epithelial cells. So it is lined by cuboidal epithelial cells having a brush bordered appearance inside them, having a lot of microvilli to increase the surface area, because as the blood is getting filtered, there are some important substances are also getting filtered out, so we need to absorb them. For absorbing them, we need a large surface area inside the proximal convoluted tubule. And for having a large surface area, you also need microvilli. But to absorb the substance, you need energy. That's why there is numerous amount of mitochondria. So the proximal convoluted tubule is lined by cuboidal epithelium, having microvilli, that is the brush-bordered appearance, and a lot of mitochondria. Now let's see the loop of Henle. The proximal convoluted tubules continues as a U-shaped structure, that is the loop of Henle. If you notice, the loop of Henle is present in the medulla, and it is having two parts. As the loop of Henle goes down, the thin descending limb, there is a thin descending limb which continues as a thick ascending limb, and there is a hairpin bend. So loop of Henle is formed by three structures. The proximal convoluted tubule continues as the descending limb, which is thin. Then there is a hairpin bend, and then it continues as a ascending limb which is thick. So this is about loop of Henle. Now let's see the last or the distal part of the nephron, that is the distal convoluted tubule. Now the distal convoluted tubule is also present in the cortex. it is less coiled compared to the proximal convoluted tubule, and inside the distal convoluted tubule there is presence of cuboidal epithelium but with no microvilli or very less or even the absence of mitochondria because there is nothing to absorb here. So that was about distal convoluted tubule, and the distal convoluted tubule continues as a collecting tubule which opens into the collecting duct. So that was all about the Malpighian body and renal tubule, and in the next module we will study about the formation of urine, and the first step of the formation of urine is ultrafiltration. Till that time, take care. Thank you.